My name is Adriana Nowak and I work at the Department of Environmental Biotechnology at Łódź University of Technology in Poland. Thank you for inviting me and for the opportunity to give this presentation, which is entitled Honeybee Microbiota, Chemical Contaminants in Honey and Probiotics for Honeybees. The honeybee plays a key role in the human environment, primarily because of its participation as a pollinator of crop plants. For several years, we can observe the phenomenon of the colony collapse disorder. CCD is a sudden death of bee colonies, and it is a global problem. There are several factors suspected of causing CCD. The main cause is considered to be bee diseases caused by parasitic mites such as Varroa destructor, viruses, and fungi of the genus Nosema. Another serious cause can be pesticides used in agriculture, mainly neonicotinoid insecticides, but also those used in apiary practice to eliminate bee pathogens. Currently, it is more and more often believed that the above factors may negatively affect the microbiota of the digestive tract of bees, which has been intensively studied only for several years. Nothing is known about many of these factors, how they work, and how they affect the honeybee microbiome. It is still under study. It turns out that it is influenced by environmental factors such as the landscape, the season, which is also related to the diet, the hygienic conditions in the hive, and the hive environment. Other factors include the type of forage and the presence of phytochemicals. Next factor is age. A worker bee lives about 35 days. But this microbiota differs especially between the larval stage and the adult worker bee. The queen lives from 3 to 5 years and has different microbiota. In general, bees tend to bioaccumulate pollutants, but also partially detoxify them. While bioaccumulation can't harm them too much, in the sense that it can hurt humans, since they live too short to get cancer, but they carry these chemicals into honey and bee products which we consume. In their short life, pollutants can affect the bee's immunity and susceptibility to infections, even with opportunistic pathogens. The gut microbiota of honeybees, this is the worker bee, is unique and relatively simple compared to that of humans or vertebrates. It is highly specialized in digestive food rich in saccharides. Nectar contains mono, di, and polysaccharides and also aromatic compounds. Pollen may contain more complex compounds, and nectar and pollen are first directed to the crop, where they are stored. Here there is an unfavorable environment. High osmotic potential is in the crop. There is no abundant microbiota. In general, the gut microbiota of bees is specialized in digesting food rich in sugars, pollen, cell walls, pectins, etc. In general, the gut of the honeybee is dominated by microaerophilic conditions with a temperature of 35 Celsius and the presence of saccharides, which is an ideal living environment for lactic acid bacteria. The digestive tract is inhabited abundantly by lactic acid bacteria and Apilactobacillus kunkei species, which is characteristic for honeybee microbiota. Despite unfavorable conditions in the crop, we can find here lactic acid bacterium Apilactobacillus concave, which dominates in this site. It forms here abundant biofilms. The honeybee microbiota is composed of core and non-core species of different microorganisms performing a number of important functions in the honeybee organism. Gillamella species and Snodgrassella alvi are specialized intestinal symbionts involved in the digestion of complex compounds from pollen. Snodgrassella alvi doesn't ferment saccharides but metabolizes carboxylates. Gillamella species ferment saccharides. The products of this metabolism are organic acids which provide a source of energy for Snodgrassella alvi. There is no antagonism between core bacteria, rather there is symbiosis and proto-cooperation. Non-core bacteria are found in smaller numbers. Frishella perrara is probably competitive with Gillamella apicola. At sites of adhesion, it causes melanization, which is unfavorable but is tolerated due to the activation of the immune system. That is why Frishella perrara is referred as symbiotic. However, it is not clear whether it is beneficial, neutral, or commensal. Other bacteria of the non-core genome, such as Bartonella, Bombella, and Apibacter species, display poorly understood functions and activities. Bombella species play an important role in the larval and queen mother development. A significant part of the queen's microbiome consists of Bartonella species, which play a role in her nutrition and modulating her fertility and longevity. The ileum of worker bees is dominated by lactic acid bacteria and the genus Bifidobacterium, and is the most abundant microbiota ecosystem in the honeybee gut, which are core bacteria. Lactic acid bacteria also dominate in the hive environment. Functions of the lactic acid bacteria are presented in this slide. These are saccharides fermentation, production of metabolites such as lactic acid and short-chain fatty acids, and other important antimicrobial metabolites which produce unfavorable acidic environment for development of pathogens. Just how many xenobiotics and honeybees and other pollinators are exposed to is evidenced by the presence of these compounds in honey and bee products, where can be found polycyclic aromatic hydrocarbons, polychlorinated biphenyls, heavy metals, dioxins, particulate matter of various diameters, pesticides residues and their metabolites, also asphalt components, and even microplastics with as yet unproven negative effect on bees' health. Example effect of environmental contaminants on honeybee microbiome are presented in this slide. Antibiotics can increase the likelihood of Nosema and Serratia infections. They lead to an imbalance in the gut microbiota system. Tillozin perturbs the microbiota, especially at the stage of acquisition. In the case of microplastic, drastic decrease in lactic acid bacteria, increase in Klebsiella and Enterobacter species was observed, especially when it was combined with antibiotics. It is known that many pesticides can poison these insects, especially non-nicotinoid insecticides, but also those used in apiary practice to eliminate bee pathogens. They can cause acute poisoning, but continuous exposure to lower doses can decrease the condition and immunity of bees, increase susceptibility to infections, and can lead to many unfavorable changes in metabolism, detoxification processes, and can cause imbalance in the proportions between individual taxa of microorganisms in gut, leading to dysbiosis. While the symptoms of bee poisoning with pesticides are better known, their influence on the gut microbiota is discovered. It was observed that pesticides significantly reduce the level of beneficial bacteria, mainly lactic acid bacteria, although the opposite results were also obtained. They can also lower the level of other core bacteria, such as Bifidobacterium species, Snodgrassella alvi, and Bartonella species. They can also increase the amount of opportunistic pathogens, such as Serratia and Klebsiella species. The research presents the results of study on the effects of pesticides such as acetamiprid, thiacloprid, and oxalic acid on the microbiota of the digestive tract of honeybees. For all three compounds, changes were observed in the proportions and numbers of each genus of bacteria compared to the control sample. The most significant changes in the pattern of the microbiota were caused by oxalic acid. It significantly reduced the levels of Bartonella, Klebsiella, and Bombella species. Bombella was practically eliminated. It slightly reduced the levels of Commensalibacter species and increased the amount of Bifidobacterium, Freeshella, Gillamella, and Lactobacillus species. Oxalic acid also disturbed the acidity of the gut, and that is why it can alter metabolism and nutrient absorption. This, what we can observe here, is called dysbiosis. Other pesticides, such as fipronil, significantly reduced bacterial levels within the genera Bifidobacterium and Lactobacillus, while increased the abundance of Gilliamella and Snodgrassella. Also, glyphosate strongly reduced the amount of Snodgrassella, partially Gilliamella, while it caused increase in the level of Lactobacillus species. Glyphosate exposure of young workers increased mortality of bees subsequently exposed to the opportunistic pathogen Serratia marcescens. In general, we observe the correlation: the higher the dose of pesticide and the longer the time of exposure, the more significant changes in the microbiota of the honeybee gut and in its body. The varied response is due to the specificity and chemical properties of the pesticide. The most common residues of pesticides in honeybee dead colonies, honey and honeybee products are listed here. It should be mentioned that pesticide residues are generally detected in every tested honey sample, even those from areas considered as ecologically clean. The concentration of contaminants is presented as follows. Honeybee accumulate more than propolis and bee pollen and more than honey. So honeybee body accumulates the most contaminants. A certain pool of them is detoxified through their own enzymes or of enzymes of the gut microbiota. So this insect is a very good bioindicator of environmental pollution in a given area. Due to CCD and increasing environmental pollution, we search for ecological methods that would show protective effects to honeybees. and an alternative could be probiotic preparations. The most commonly used in probiotic products and preparations are lactic acid bacteria from the genera Lactobacillus, Lactic-Aceiobacillus, Lactoplantibacillus, and Lactobacillus. These bacteria naturally inhabit the bee environment, such as flowers, bee products such as honey, royal jelly, plant silage, as well as the digestive tract of humans and animals, including the honeybee and other pollinators, where they perform beneficial functions. This slide presents a summary of research on the effects of probiotic bacteria on honeybee health. The search for strains of lactic acid bacteria with specific beneficial health-promoting properties is intensive, long and expensive, and the effects must be confirmed not only by laboratory tests, but also by semi-field and field studies on bees. So both the strain and the preparation must show scientifically documented efficacy. It is worth emphasizing that a particular feature of a strain, for example, protection against Nosema cerana infection, is specific to an individual strain of lactic acid bacteria, not to particular species, genus, or all lactic acid bacteria. So, a probiotic strain must exhibit health-promoting effect, must have a specific effect over and above that manifested by all lactic acid bacteria. Supporting honeybee organisms with probiotic bacteria can be used for biocontrol of some honeybee diseases. The use of probiotics in the detoxification of pesticides, including neonicotinoid insecticides, is also promising. From 2019, our department is implementing a project entitled Selection of Microorganisms for the Construction of Ecological Protective Preparation for Honeybee, Apis mellifera, of which I am the manager. This presentation was prepared within the project. Thank you, my team. Thank you, the organizers. Thank you very much.